Okay, I'm going to introduce Irving here. Dr. Irving Vega has been an associate professor in the Department of Transactional Neuroscience at Michigan State University College of Human Medicine. His research focuses on molecular biochemical mechanisms that modulate the accumulation of pathological tau proteins in Alzheimer's disease and related dementias. Vega earned a BS in biology from the University of Puerto Rico and a PhD in the Department of Cell Biology and Neuroscience at Rutgers University. In addition to all that, he also has some other credentials here. I'm going to have to get it right. I noticed it also says up on the board, the Red Cedar Distinguished Associate Professor. This is an award given periodically to up-and-coming distinguished associate professors at Michigan State University. He achieved his in 2023. So he's also the current director of the Integrated Mass Spectrometry Unit in the College of Human Medicine at Michigan State University. And he's program director of Endure, a bridge to the PhD in neuroscience program at Michigan State University. So without further ado, Dr. Irving Vega. Thank you.
Well, thank you everyone for being here. It's an honor and a privilege to have the time to talk to you about something that I'm really passionate about it. Been studying Alzheimer's disease and the pathological process for the past 20 years. And it's something that always amazed me, trying to understand not only the pathological process, but also how to reduce our risk of developing this disease. So today, my goal is to go through the different aspects of Alzheimer's disease, from the molecular aspects to health disparities. And at the end, talk about how we can reduce our risk of Alzheimer's disease. It's never too late to reduce our risk of developing Alzheimer's disease, but it's a disease that actually we need to, in order to reduce the risk, we need to start changing how we see this disease. It's not a disease of the elder. It's something that we tend to accumulate since we are born, and how we are raised, the environment that we are in since we are a child, all the way to adolescence, young adulthood, and then adulthood. So we need to, the first stigma that we need to eliminate is that this is a disease of all people. It's not. There are people that will have the pathological hallmarks that I'm going to mention today and never present symptoms. So there are things that we can do. Okay, move forward. Okay, it's not moving forward. We have the first technical program here. Let's see how we can resolve this. Okay. Okay, there we go. Let me see if I can put back the laser and it still move. Okay, perfect. And then I can. Okay, there we go. Perfect. So, yeah, so technology always is good when it works, right? So some of my conflict of interest declaration, I don't have any financial conflict of interest of what the things that I'm going to mention. And I serve in different boards, and my opinions here are my own. It's not working again. There you go. Let's see if now it works. Okay, good. So dementia is a umbrella term that is used to describe when someone is presenting problems associated with malfunction of the brain. And it could be behavioral, it could be cognition, it could be executive function, it could be reasoning. And it's a term that sometimes the physicians use when they know there is a malfunctioning in the brain, but they still don't know what type of dementia is. And this is one of the first questions that I always receive. Is Alzheimer's disease, what is the difference between Alzheimer's disease and dementia? Well, what happened is that Alzheimer's disease is the most prevalent type of dementia, and is the most known, and sometimes the most diagnosed incorrectly, because the symptoms associated with Alzheimer's disease tend to overlap with other types of dementia. And depending on the presentation, the symptoms, the first symptoms that were noticed by the person living with dementia or the family members, it can take the physician that is looking at the person living with dementia in a different route. And it's because all the time he's trying to exclude other things to get to the diagnosis. Sometimes it gets confusing. And the other types of dementia that can be confusing and misdiagnosed as Alzheimer's disease is vascular dementia. Vascular dementia is, as like the name suggests, is associated with problems in the cerebrovascular, in the cerebrovasculature. Our brain is highly have a match of vessels all around the different areas of the brain. And this mass that is of all cells and lipids that we have, that we call brain, all the blood and that carries oxygen and nutrients have to reach all these different areas that are very deep in this mass of cells and lipids, okay? So there are very thin capillaries and veins, and those very thin capillaries and veins sometimes break, or some things like lipids accumulate inside. And you start getting micro strokes or strokes. And sometimes a stroke could be as someone pausing and then continue. That could be a micro stroke, okay? And the accumulation of those strokes tend to limit the oxygen in that area where the stroke took place. And the cells around that area, because they don't have now oxygens or nutrients, will die. And as more cells continue to die, then clinical presentation, depending on where that is happening, is the clinical presentation is when symptoms happen. But that takes years and years, because our brain is a magnificent machine that when you have something wrong in it, it will repair. It will try to repair. And if it cannot repair, just go like we do during this season that is finishing in Michigan, construction. You know, we have 2 seasons, winter that is coming and construction that people are rushing now to finish things, that they have detours, right? And then now, before it took you 15 minutes to get to point A to B, now they have a detour because the engineers are very, you know, they're very good about getting detours, and now it takes 20 or half an hour. The brain can do detours, and then you can facilitate that new connection and go around that area that was damaged. So the other one is frontotemporal dementia. And frontotemporal dementia is characterized by behavior changes, lack of inhibition. It will be someone that is feeling hot right here in this room, will take the clothes out and feel fine about it. I was hot, so I just took my clothes off. And that's it. Oh, I'm talking here, and the person will stand up and start cursing. Is no inhibition, the person don't realize that are doing that. Usually that's the first symptoms associated with frontotemporal dementia. And then you have dementia with Lewy bodies. And the characteristic of dementia with Lewy bodies is hallucinations. The person have vivid hallucinations, acting hallucinations. They will see someone and they will fight, and they will try to, if they feel threat, they will attack whatever is in front of them because they're seeing the hallucination. And one of the problems with that is that the reality get distorted, and the wife or the husband will be the first subject that will get attacked. And they really leave those hallucinations. And that is one of the characteristics of dementia with Lewy body. There are other ones where I just giving you the distinction. Because you know what is the distinction factor for Alzheimer's disease, which is memory. And that is one that is usually detected secondary sometimes, but sometimes is the primary or the chief complaint that take someone to see a neurologist, which is that the person don't realize that are repeating themselves about what they say 5 minutes ago, or they forget how to get from the house to buy grocery to the same grocery place that they used to go every day. Or they get to the grocery and buy the groceries and then get out of the place and forget how to get back home. So those aspects of execution, executive function, making decisions, and also memory, short-term memory, are the first chief complaints that will take someone to see a neurologist. The caveat with that is that we know that when clinical presentation happens, there are millions of neurons that already have died. And unfortunately, when those neurons die, there's no way back. And that's what happened to Auguste D. Auguste D, I'm gonna go a little bit into history of Alzheimer's disease and why we call it Alzheimer's disease. So Auguste D, in her early 50s, which by that time, this is 1894, 95. At that time, 50, being the early 50s, it was a big achievement. It's like 85 now. And her husband took her to this hospital where Dr. Alois Alzheimer was the main physician under the mentoring of Dr. Emil Kraepelin. And he starts seeing Auguste D and noticed that she was completely different. And the famous quote from Auguste D that one time told Dr. Alois Alzheimer, and he made a note of it, is, "I have lost myself." Some days she was completely fine, normal, and then the next day, lost. Didn't know who she was, where she was, where she were. It was a complete different person. This is the picture that we use about Auguste Dieter. Actually, the D is for Dieter. But it's not representative of someone living with Alzheimer's disease. The diagnosis of Alzheimer's disease, someone that is diagnosed with Alzheimer's disease, can be functional for 5, 10, up to 20 years, or even more in some cases. Is something that we need to take also out of our minds, that a person diagnosed with Alzheimer's disease will look like that. There's still things that can be done, and there's some things that need to be adapted, but the person is still functional for 5, 10, depending on the individual and the other comorbidities or diseases that the person has. So once she died, because he was also a neuropathologist, he took the brain, did an autopsy, and a study, and used, in Germany, there were these amazing techniques using silver to stain the brain. And he found this pathological hallmark in the brain, and then presented that in a symposium. And then Dr. Kraepelin started fomenting or proposing that this disease that Dr. Alois Alzheimer presented in this conference need to be called Alzheimer's disease. But the thing is that he was not the only one working on this type of dementia of patients that were presenting with this lack of memory and cognitive functions. And one of them was Solomon Carter Fuller, an American, African-American, that actually went to the same hospital where Kraepelin and Alzheimer was there. And actually he was working under Alois Alzheimer. And he was the one that did all that staining. And then he came back to the United States and he identified cases in the United States, similar to the case that they identified in Germany. Unfortunately, because of the time, he was highly discriminated and he was not able to present the cases that he was actually discovering. Even though he managed to get a publication about it and presented more cases that Alois Alzheimer, he was completely shut down from the scientific community, and we are not calling it a Fuller disease today. There was another guy that actually was in a competitive in the competition and in a hospital that was in competition with the hospital that Alois Alzheimer had, and this guy Oscar Fischer was under the mentorship of Parkinson, Dr. Parkinson disease. We're not going to talk about Parkinson disease, but if you have questions about it, we can also talk about that. But Parkinson then was guiding Oscar Fischer, Dr. Fischer. But unfortunately, was at that time in the 40s, in 1907, he published actually an article that had about 16 cases. Again, more than the cases that Alois Alzheimer presented. Unfortunately, he was killed in one of those concentration camps in the 1940s because he was a Jewish in the Czech Republic. So it's not Fischer disease. And we call it now Alzheimer's disease. That's a little bit of the tragic history behind what we call today Alzheimer's disease. Today, we know that Alzheimer's disease in the United States is close to or more than 7 million people that are being diagnosed with this disease. And we know that there are more cases out there because it's an estimate that about 50% of the cases of Alzheimer's disease are either misdiagnosed or not diagnosed at all, that people don't receive a diagnosis. They receive a diagnosis of dementia, and I bet you guys have heard that before. I know someone that have dementia, and it's because the physicians don't know yet and don't want to declare as an Alzheimer's disease. There is a recent paper that looked at different areas in the United States for the prevalence of Alzheimer's disease. Michigan is right there in terms of the number of people living with dementia. This is a work published in the Alzheimer and Dementia Journal, Journal of the Alzheimer Association. And Michigan is right there, although this number is still unclear whether that actually is the amount of cases that we have and the prevalence that we have, because again, the misdiagnosis and misdiagnosis and no diagnosis of the disease. So we are going to be facing a problem soon because we also don't have sufficient neurologists, sufficient psychiatrists, neuropsychologists. We have a shortage of about 1 million to address more than 1 million people a year that can go to a different hospital around the nation. So it's something that we need to take care of it. So what is Alzheimer's disease and what was discovered by Fuller, Fischer, and Alois Alzheimer? It's the accumulation of a piece of a protein, a peptide. Proteins are the functional aspect, are the workers in the cell. And we have the map of the instruction, which is the DNA. Our genetic material is the information, which then is transcribed, is read out, and produce an intermediary molecule that is called RNA. And that RNA molecule gets out of the nuclei, of the nucleus that have all our genetic material in the cell. That mRNA get out of the nuclei, and then inside the nuclei, but still inside the cell, that message is transcribed into a protein. And the proteins are the workers, are the ones that do all the functions of the cell, the metabolism, the signaling, the response of the cell. If you are cold today or you're hot today, that's through the function of protein, stimulations of proteins inside the cell that actually are doing the work, okay? So the piece of a protein gets cut, and I will show you later on what it is, and accumulate outside of the cells. The main cells in the brain that carry this function and communication are called neurons. And these neurons are the ones that produce this peptide that is aggregated outside of the cell. The other pathological hallmark that Alois, Alzheimer, Fuller, and Fischer identified was another protein accumulating inside the neurons. So the peptide that I was mentioned before, we call it amyloid beta peptide, accumulate outside, but the protein that accumulate inside that Bill mentioned that is what I have been working is the protein tau. And these protein aggregates form aggregates that I'm going to show you in a second inside the neurons, okay? The combination of these two pathological hallmarks is what define a brain with Alzheimer's disease. You have to have these two pathologies in order to confirm that the diagnosis that the neurologist did is correct. Because unfortunately, right now, the diagnosis of Alzheimer's disease is probable or possible Alzheimer's disease. There is not a single test right now that you can do to say you have Alzheimer's disease, you have vascular dementia, you have frontotemporal dementia. We don't have that yet. But because of the clinical presentation, how the disease presents and progresses in the individual, we can say whether it's probable or possible Alzheimer's disease. And then these pathological hallmarks start accumulating in a specific brain regions. In the case of Alzheimer's disease, it's in the temporal region, specifically in the limbic system. A big word to say the center of learning and memory. That's what it is. And that region that is formed of different small structures inside of the brain, these pathological hallmarks aggregating those cells, start killing those cells, and that pathological hallmarks tend to then spread throughout the brain. And that's how the progression of symptoms and clinical presentation of individuals from having some mild cognitive impairment, some mild problem with memory to completely memory loss, of not remembering even their own names, the family where they are, and all that. So how these pathological proteins throughout the years start spreading at different regions. The frontal cortex, right here, this is the front of the brain, right here. This is when behavioral problem starts. And this is the region that is first affected in frontotemporal dementia, but in Alzheimer's disease, behavioral problems tend to affect later. But in some cases, behavior problems like anxiety, depression are presented as well in Alzheimer's disease. And then it continues spreading throughout the brain until you have then sensory and motor deficiencies or dysfunctions. And then the person end up being bed bound and then die of lung problems or kidney problems or heart failure, okay? So what are those? Because of the accumulation of those two pathological hallmarks, the main hypothesis in the field is called the amyloid cascade hypothesis, which that amyloid cascade hypothesis states is that the accumulation of those amyloid beta peptides that I mentioned that aggregates outside of the neurons, these start first and send signal to the neurons and the other cells in the brain that are the glia cells. Not going to get into that detail, but those glia cells. And then those signals that are induced by the accumulation of that peptide, then the neurons start reacting to it and start then affecting the tau protein, which accumulate inside the neurons. And that tau protein is the one, when accumulated inside of the neurons, kill the neuron. Okay, so it's mediated by the accumulation of A beta, but it's actually executed by tau, by the accumulation of tau. To go more molecular, and I don't want to lose you here. This is the way that peptide is produced. I told you that proteins are the one that does carry job and do the work in the cell. We still don't understand what the amyloid precursor protein, what really is the biological function of this protein. We don't know what it is. We know it's important, but we don't know exactly what is the function of this protein. There's some speculation about it in neuronal development, signaling, synaptic plasticity, which is the communication between the neurons, but it's not very well understood. What we do know is that when this protein that is in the membrane of the cell has this lipid membrane where this protein is intercalated there, and then when it's intercalated there, the protein then get cleaved by other proteins. Other protein cut this amyloid precursor protein, generating this amyloid beta peptide that then goes outside of the cell and aggregates. And the difference between the amyloidogenic pathway and the amyloidogenic pathway is just two amino acids. I mean, two amino acids are what form the proteins. And the difference between A beta peptide that is 40 amino acid long versus 42 amino acid long is what make this peptide more to aggregate, just two amino acids. So these proteins here, I want to emphasize or make attention to presenilin 1 and presenilin 2 is a complex of protein that actually mediate that cleavage. And when that cleavage is made, then that peptide is actually secreted outside and not only affect the communication between neurons and establish or initiate signalings that affect the protein tau, but also get intercalated in our vasculature. And once it gets into the vasculature, it can actually block it and you can have a stroke. The thing is that if it's not detected, you guess probably right that then the diagnosis will be vascular dementia. Because now the doctor do an MRI and see signals of a stroke in the brain and say, oh, it's vascular dementia. Well, not necessarily. It could be that A beta in that case has been produced and accumulate in the vasculature, blocking the vasculature in something that we know is cerebral amyloid angiopathy. And if you have questions about it at the end about the current approved treatments for Alzheimer's disease, this is the main risk or side effect of the treatment that have been approved for Alzheimer's disease, this infusion of antibodies, because those antibodies get those peptides and take it where? To the vasculature to clear those peptides out of the brain. The problem is that it's already aggregated and tend to aggregate in the vasculature. And then that's why this main side effect of the current approved treatment for Alzheimer's disease is brain bleeds. So it's good, but we are not there yet. The other pathology that I was mentioned and my favorite protein is tau, although I have another protein that I'm going to show you soon. That is my favorite one. Tau is a protein that actually helps the structure of the cell. We have what we call a cytoskeleton, the skeleton of the cell. And this protein, main function, it have other function, but the main function of this protein is actually to maintain the skeleton of the cell. So when it's affected, when it's detached from the skeleton, now it's like affecting the bones of the cell. And now that skeleton starts to deteriorate and break apart. And now the cell that is required as a skeleton, now it collapses. And it's imagine my arm without bones inside, right? It will not stay like this. It will, when you have the bones, your bones broken, it's actually the skin will not hold, right? The muscles will not hold. And that's what the tau does to the cytoskeleton, to the skeleton of the cell. But once it's detached, then tends to aggregate. And that's the problem, that the protein go from being a single species to then form 2, 3 together, 4, 5, and then those proteins become and form oligomers. And from oligomers goes to filaments, and from filaments go to tangles. And the tangles was what Fischer, Buehler, and Alois Alzheimer identify inside the neurons. But now we know that the toxic effect that this molecule exerts in the neuron is actually before it gets to tangles. It's at the oligomeric or filament form. And it's because this tau protein, in a mechanism that we still don't understand, when it aggregates, for some reason, the neurons start transferring this pathological molecule to neighboring neurons. And now once it gets to the next cell, to the neighbor cell, it's released inside and then promote the aggregation of tau in that cell. And then now that cell generate these oligomers that then are passed to the neighbor cell. And then in that way, it continue killing neurons that are connected in the same brain region. And that's the image that I show you, how it continue progressing. That brew in the brain, that brew that was in the brain, continue progressing is because this tau is being, through that connections of neurons, started spreading from one neuron, from a sick neuron to a healthy neuron that now becomes sick. And now that sick touches the healthy neuron and make that other neuron sick, and then it continue, okay? That progression of the disease. So looking at my interesting, and this is my favorite protein, which is called EFHD2, I discovered this protein associated to pathological tau, and this was using an animal model that we can humanize. These are mice that we can humanize expressing the human tau protein and induce the same pathological hallmarks that we see in human brain. We can replicate that in a mouse. And by studying that in the mouse, I was able to identify this protein associated with the pathological tau that is here, called EFHD2. And this protein is, the mouse protein is 93% identical to the human protein. And we found that in Alzheimer's disease, this protein, the abundance of the protein in most cases of Alzheimer's disease is increased compared to normal or cognitive unimpaired individuals. So somehow this protein, the gene of this protein or the protein, the abundance of it increases as the pathology is presented in the individuals. And we actually look at human brains and found that this protein EFN2, this antibody PHF1 recognized only pathological tau. So we found that this protein, when it's yellow, means that they're together. So we found that this EFHD2 protein co-aggregate with tau in the brain of someone that have died of Alzheimer's disease. So now our study has been, what the heck this protein is doing? So now we went to the bench and look at the biochemistry of this. And we have human tau and the HD2 protein, and we mix them together. And we know that we can induce filaments of tau. There's ways that we can actually induce what we see in the neurons in the brain, we can induce it in the test tube. So when you add that inducer, you go from no aggregates or no filaments to filaments of tau. So then we decided, okay, let's add EFHD2 to it. We call it EFN2 as well. So what we saw is that when we added without the inducer, it already starts forming filaments. And when we added when they're tau filaments, it actually formed tangles, similarly to the structure that we see inside the neurons. So we now develop an animal model, a mouse that expresses the human tau, and we knock out the gene. And what we found was that now this animal is sicker. And what happened is that this tangle formation is actually not detrimental to the cell. It's actually a neuroprotective factor. When you accumulate these high structures, the cell cannot transmit it to the next cell, get trapped inside the neuron. By then eliminating EFN2, now this aggregate cannot be formed, and only these small oligomers are formed, which are actually transported to the other neurons. And that then tend to accelerate neurodegeneration in the animal. How that translates to humans? maybe 10 years from now or before, hopefully, but we will one step at a time. We go from animal models, we call it preclinical research, and then we can translate that later on to humans. But that's where we are. That protein, we were the first one. Our team was the first one to identify this protein in the brain. And that's my favorite protein that I have been working for 15 years now. So genetics of Alzheimer's disease. Most cases of Alzheimer's disease are what we call sporadic or idiopathic in nature, meaning that there's not a specific mutation that passed from one generation to another generation in a family, within the family, okay? Those are more than 95% of the cases of Alzheimer's disease. And they are also called late Alzheimer's disease because they actually tend to, people with this sporadic Alzheimer's disease tend to present symptoms after 65, usually after 70 years of age, okay? And it's most of the cases. But there are less than 5% of the cases where the grandmother, the father, the daughter, or the son, generation after generation, and multiple people within the same generation have Alzheimer's disease. And that is due to mutations in presenilin 1 or presenilin 2 or the amyloid precursor protein. Remember, the amyloid precursor protein is the one that generates the peptide that aggregate in the extracellular space outside of the neurons. And I'm repeating that because Bill told me that there is a quiz, there is a test after this talk. So those on Zoom, we're gonna put it in for you in the internet there to complete it. So presenilin 1, amyloid precursor 1 is the one that generates the peptide. Presenilin 1 and presenilin 2 is that macromolecule that cut APP. So that's related. Actually, the first mutation associated with Alzheimer's disease was identified in the amyloid precursor protein, in APP. Actually, the scientists, the geneticists in the 1990s found this gene actually stored in the brain of people with Down syndrome. Down syndrome is a trisomy of the chromosome 21. And individuals with Down syndrome, when they are 35 or older, they develop dementia. And when they look at the brain of individuals that are 35 or older, that develop dementia, Down syndrome, that live with Down syndrome and then develop dementia, they found the same amyloid plaque that are found in Alzheimer's patients. And they were like, wait a second, how is that possible? So they narrowed the search to chromosome 21, and that's where amyloid precursor protein is. That's in a chromosome 21 is where the amyloid precursor protein is. So these individuals with Down syndrome, because they have 3 chromosomes of chromosome 21, they overproduce the amyloid precursor protein, and they generate these peptides that aggregate later on in life, and they develop dementia. And that was the first mutations that were identified associated with Alzheimer's disease. But the most mutations in families is in the gene presenilin 1, one of the ones that cut the amyloid precursor protein. And actually, in the lab that were Alois Alzheimer, they found slides, tissue in a slide that they were looking on the microscope, and they scrapped the tissue and extracted the DNA, and they found the Augusti Dieter had a mutation in presenilin 1. So most of the cases at that time, in the late 1890s, early 1905, 1906, those individuals most likely had a mutation, were early onset, because one of the aspects of early onset of mutations in this gene is that individuals develop the clinical presentation associated with Alzheimer's disease in their 50s, sometimes in their 40s, and their cases even in their 30s. They start developing cognitive impairments. And especially are detected in individuals in their 30s when they have mutation, because the the physician know the history and they start checking for it. And they start finding this cognitive impairment early on. So that's why we can divide Alzheimer's disease in two types, late onset or early onset. And in early onset, it's most likely to a mutation that goes within the family, okay? And multiple individuals within the same generation, okay? But there are other genes that are known as risk factors. So the main genes that are associated with causation are presenilin 1, presenilin 2, and APP, but there are a series of genes that have been found associated with Alzheimer's disease. One of the main risk factors for Alzheimer's disease, especially within the white population in the United States, is the ApoE4. And ApoE4 is actually a gene that was first known for its association with cholesterol and how it managed cholesterol and help transport cholesterol, because cholesterol, despite of what we think, our cells need cholesterol. So low cholesterol is bad. Too high cholesterol is also bad. Always about the streams, right? So ApoE, the most people have ApoE3, but those people that have ApoE4 have a 50% risk of developing Alzheimer's disease, meaning that that person has 50% higher risk than someone that have ApoE3 or ApoE2, okay? And I want to make the emphasis on risk, because if you have ApoE4, that is rare, but you might have one copy or two copies of ApoE4. You have two copies of ApoE4, is higher your risk, but it means that you are a higher risk. It doesn't mean that you're going to develop Alzheimer's disease, okay? But if you have ApoE4 with these other genes that have been associated with the disease, now you start getting a combination of genes that then in the future, that will increase your risk in addition to other things that I'm going to talk later on. So, but we are learning now that even having a mutation in presenilin 1, which I told you is associated with Alzheimer's disease within families. And before this paper, that was like 2 years ago, we thought that if you have a mutation in either presenilin 1 or presenilin 2 or amyloid precursor protein, it doesn't matter if you live in a bubble. If you have a mutation in either of these three genes, you're going to develop Alzheimer's disease. But we found a group led by Jaquel Quiroz at Harvard University that are following a family in Colombia that have a mutation in presenilin 1. They found this individual right here that has the same mutation similar to her family members, but don't develop cognitive impairment. Actually, her family members were developing cognitive impairment in their early 50s. She's already 75 years old and is only mild cognitive impaired, having the same mutation that the other family members have. When they decide, okay, let's sequence the genome, all the genes of this woman to see what is happening. And they found a mutation in ApoE3 that actually seems to affect the penetrance or the expression of the disease-related aspects of the mutation in presenilin 1. How? Is under investigation. We don't know. But what we know then, and we change, is that if you have a mutation in presenilin 1, in presenilin 2, and APP, we have to look at other genes because you might not develop the disease like other people. They're a combination of genes. And these completely provide more evidence that we need now to rethink the amyloid cascade hypothesis, because there are other pathways that are actually involved in this process. So we're looking at all these other genes now. I'm not specifically, I'm more looking at the biochemistry of tau and how EFHD2 affects tau, but other people are looking at other genes to see if they can find these genes that they can stimulate or they can reduce to then affect the progression of the disease. And that's where the research is going. In addition, just to make a parenthesis, the interaction between the environment and our genes. Since we are born, the environment where that kid, that baby is born, is already modifying his or her genomic material in a process called epigenetics. So in the environment, we get these stimulations, and that goes to the gene and change the material that is inside the nuclei and affects the expression of the genes. So it's affecting the music. And let me give you an analogy. You go to a concert, opera or a symphony, right? And they have the same instruments. But you can, when they start playing, the first act, you can distinguish that act from the second act, right? It's the same instrument, but they play different tones. And with those tones, develop a new song. That's us. That's why we have 99.9999999% of the genes are the same. Bill and I have the same genes. For his son is different than my son. And then that's why I look different than Bill. But his genome then is more similar to your son or daughters or family members, and then you look more like them, okay? When the environment is interacting with the genome, it's changing the song. And sometimes there's gonna be one violin back there that one chord got broken. And we probably don't gonna notice because, I'm not musician, so one violin might not be enough for me to detect that there is a problem, but there is 2, 3, 4. Now you start seeing that there is a problem. It doesn't sound like it's supposed to sound. And that's where disease happens. When you have multiple genes that have been changed, and then the song now doesn't sound the way that's supposed to be sound. So because we know these pathological hallmarks are in the brain, there have been developed specific radioisotopes that are injected in the individuals, and these radioisotopes goes all the way to our blood, to the brain, and are specifically binding to these protein aggregates. In the case of amyloid, the amyloid peptide, this pister compound B, and there are different forms now, aggregates or actually interact and gets accumulated where the peptide is. And what you can see in a normal individual versus someone that has been diagnosed with Alzheimer's disease, the red indicates where the peptide is. This FDG is actually an analog of glucose. So when it's injected and the brain uses it, now produce radiation and can be detected in a PET scan. So when you're using it, you have more signal. That means that there's cells that are using that analog of glucose and they're active. When you don't see red, it means that there are no cells using that glucose. So what you can see here is that reduction of the signal of ADG coincide with increased signal of the A beta, meaning where you have a beta peptide, you have less cells. But this is not what we see typically. This is something very, a selected case. It's not that simple, okay? We can do the same thing with tau, and there are radioisotopes that now combine tau, but the thing is that tau not only aggregates in Alzheimer's disease, tau also aggregates with other neurodegenerative diseases like corticobasal degeneration, progressive supranuclear palsy, chronic traumatic encephalopathy, the one that happens to the football players, or used to call dementia pugilistica for the boxers, the pathological molecule associated there is also tau. So these markers can bind tau in different brain regions, and in that way, it can be distinguished between Alzheimer's disease, CVD, and PSP. But again, these are very textbook cases. It's not as simple. What we are also moving to is actually to look at biofluids, either in this cerebrospinal fluid. It's a spinal tap. It's painful. It has side effects. Some people don't like to do it. That's why we are now concentrating more in blood. But you can actually detect tau and A beta. In the cerebrospinal fluid, a decrease of A beta means that it's getting accumulated inside the brain. An increase of tau in the cerebrospinal fluid means that pathological tau is being accumulated in the brain. So we can use that also to detect in blood, but it have not been sufficient. The specificity and sensitivity of these markers are in their 80%. And that means that I have a lot of way to improve. So what we have been doing in the lab is actually doing quantitative proteomics, which is a way to take all the proteins in the blood, process it, use an instrument that is a mass spectrometer, and then collect all information and then look at the abundance of these proteins and then decide, okay, is increasing someone with cognitive impairment or increase or decrease compared to cognitive normal or cognitive unimpaired individuals. And in that way, develop a set of proteins that can help us in the diagnosis of Alzheimer's disease or cognitive impairment, in addition to tau and A beta, okay? So we have been doing that using, taking blood from individuals that have mild cognitive impairment. Why? Because we want to identify that there is a problem in the brain before they have full-blown dementia. Because once they have full-blown dementia, there's no way back. But if we capture them when they have mild cognitive impairment, there's things that we can do to delay that progression, okay? We have identified several proteins, and now our idea is to develop this bioinformatic tool that when you go to the doctor and you have different diseases that are being associated as risk factor for Alzheimer's disease, we can then take that into an algorithm and decide how much risk you have to develop dementia, okay? So an idea is to have, okay, you have obesity, you have metabolic disorders, and you have one of these proteins that we identify, but not cognitive impairment, then you have a low risk. Or you have just a mild memory problem, then you have low risk of going into dementia. If you have now memory and executive function dysfunction, and you have hypertension and 3 proteins of this one that we identify, now you have moderate risk. And also you have A beta and tau. But if you have all of this, all the domains in cognition, plus all the proteins that we identify, plus A beta and tau, now you have high risk to go from mild cognitive impairment to dementia, okay? That's where we want to be soon, hopefully. We'll see. We already identified some proteins. We're working with biostatisticians to actually develop this kind of algorithm that when you get the blood work, we can put that in a system, and the system the output to the doctor will be to tell you are a low risk, you are a moderate risk, or you are a high risk to transition to dementia. Because if you are diagnosed with mild cognitive impairment, it's not necessary you're going to go to dementia. Okay, if you have mild cognitive impairment, not necessarily you're going to go and develop dementia. We want to distinguish that. We wanted to distinguish who is actually a higher risk versus the other ones. And then in that way, we can then reduce that burden that we have of people waiting for diagnosis or treatment or things like that, right? We can actually prioritize those that are from higher risk, from low risk in the system. And then instead of seeing it every year, then we can see those every 5 years. And then the ones that are higher risk, then see it more often. And in that way, we can spread and affect the flow of people going to the clinics. But Associated with Alzheimer's disease, there is health disparities. And we know that women are at higher risk of Alzheimer's disease than men. And African Americans are also a higher risk, and Latinos are also a higher risk. And we know that we can do a lot of things to reduce these health disparities and go into health equity. One of the problems is access to healthcare and discriminations in the system that affects African Americans and Latinos, and the intersect of that. Because imagine a woman, African American, that now has a double risk as a woman and also as an Afro-American. Same thing with Latinos. So what we know in terms of risk factors that contribute to these health disparities, the number one is education, which is a proxy for many things, right? So you lower low education level, that means tend to be low income, which tend to then live in areas that don't have that much amount of access to healthcare, right? And what I want you to notice here is the yellow one is cognitive impaired and the blue ones are normal cognition. You can see that all these risk factors, low education, low income, and rural residents, affects regardless of the ethnicity. You see that it is higher, the percentage. It affects everyone. It's the magnitude of it that is the difference. The disparity is that it affects more. It's not that don't affect white, is that the proportion is higher, but these risk factors apply to everyone. And that's another message that I wanted to bring today is one, it's not a disease of all people. It is something that we need to do to our life. And if you don't have cognitive impairment, or even if you have mild cognitive impairment, there's still things that you can do. We know that there are specific mutations. It's not only about genetics on the Latinos, for example. I identify a mutation on presenilin 1 that is only in Puerto Ricans. I'm from Puerto Rico, and this mutation is only migrates among Puerto Ricans, and they develop Alzheimer's disease. But like I mentioned before, even though if you have the presenilin 1, if you have ApoE3, it actually affects different in Afro-Americans. And we talk about the Christchurch mutation that in the Colombian family is protected, right? Okay, so for cognition, this is just, again, that is not about ethnicity, it's not about genetics. This is Latinos, immigrants, Latinos that were born in the United States versus Latinos that migrated at younger than 18 years, between 18 to 34, or after 35 years of age. And TICs are a metrics of measuring cognition, and this is a baseline. When we correct for age, sex, and education, Latinos that migrated to the United States, regardless of the age that they migrated, they have the same cognitive function as white. But something happened to the Latinos that are born in the US, that the cognition was lower. And that was regardless of the domain of cognition. It was the same. So it's born here what made the difference between Latinos in terms of the cognitive performance at baseline. So what that means, we told you that we have this amyloid aggregation, we have tau pathology, and that leads to neurodegeneration, right? So this is what is all behind that. We have, and this is actually called the health disparity framework, which actually is a vicious cycle of disparities where you have environmental factors that affect your genes. You then, because of lower education, you get impacted by sociocultural factors, lower education in areas, as you know, here in the United States, the quality of the school, it depends on your zip code because of the property tax associated with that region and the resources in that school. So you are in a low income neighborhood, the resources at that school are lower than in a higher income school. and that create disparities. Is how our brain gets connected from when we are a child all the way to adulthood. And once you then start accumulating all these risk factors about where you live and your sociocultural factors, that has an impact in your biology. It's changing your biology. And once it's changing your biology, now you're increasing comorbidities to have hypertension, diabetes, obesity, higher cholesterol, which then continue affecting your biology, increasing inflammation, which then leads to metabolic, I mentioned metabolic disorders. And then you combine that with our system getting lower, repairing our brain. Now you have the recipe to then develop neurodegeneration, have a higher risk of neurodegeneration. So what can we do? You know, I have friends here that likes to dance salsa, and social dancing, very important, okay? Being here, learning something new, socializing, being out there, working with others and in learning a new task, regardless of what it is, maintaining your brain active is very, very important. Either puzzles, anything, even if you would like to work on your yard, perfect, do that. Exercise, walk. You don't have to be a marathon runner. That's not what exercise means. Exercise means to maintain your system active, your metabolism. Because if you move your blood, that's actually getting into your brain and cleaning those toxic molecules that accumulate in the brain that can lead to the accumulation of A beta and then tau, okay? And if you are doing exercise, your heart is working, and what is good for your heart is good for your brain. And also for what is good for your stomach is good for your brain. Food is very important. Have your meals, reduce the amount of high processed food is very important. Try to eat green leaves, a glass of wine, a small glass of wine. I always have to emphasize that, right? One small glass of wine, because those fruit that have a dark color skin have antioxidants that are good for your brain, okay? Green leaves is the same thing. Everything, any vegetable that is green, that have green leaf, that are associated with green leaves, good. Avocado, great, the best one, okay? Fish, be careful from where you get your fish, especially here in Michigan, that are our rivers and lakes, some of them are highly contaminated with PFAS and PFOS, which actually is really bad for our brain. And is the reason why Michigan and all others like Wisconsin and Minnesota, this area here, is had the highest prevalence in the United States of ALS, or Lou Gehrig disease, which is a neurodegenerative disorder that affects the peripheral nervous system and then the motor functions in the brain, okay? And it's because of the contamination that we have in the lake. So be careful, you know, we are all about the Mediterranean diet, but you have to be careful where your fish is coming from, because of mercury, because of PFOS accumulate in the muscles of fish. So we do a lot of things good that are in our own diets. You just have to watch your calorie intake and the quality of the food that you're getting. Farm markers, there is always one really good here in Holland that I like to come with my wife and daughters. And that farm marker is really good. You can get products there that are really good for you, okay? Hypertension, regardless of all that, if you have already diagnosed with hypertension, diabetes, high cholesterol, that doesn't mean that you're going to develop Alzheimer's disease. That means that you have a higher risk if you don't control it properly. We know that people that have Alzheimer's disease and also are diabetic, the ones that don't treat the diabetes progress faster in neurodegeneration than a diabetic that control the sugar level in their blood, okay? So even people that have already diagnosed with Alzheimer's disease, if you control your sugar level, your progression is going to be less than someone that don't control the sugar level. So keeping active, thinking about stress, and one last thing, sleep. When you sleep, your brain, there are membranes in the brain that get open. And when that you get to that deep sleep, that those membranes open and it flush all those toxic molecules from your brain, go to your bloodstream, go to the liver, and then when you wake up, you have to go to the bathroom, right? But you need to get to that deep sleep, okay? It could be, I know, anticipating a question, how much I need to sleep? Everyone is different. It could be 4 hours, it could be 5 hours, it could be 8 hours. The range is between 7 and 9, okay? But everyone is different, okay? I tend to sleep about 5 to 6 hours a day. And I know that if I hit lower than 5, I'm not that functional the next day. And you feel it in your brain. You feel that, how heavy it is. And the one of the worst torture that was applied is maintaining someone awake for more than 72 hours. The person start developing symptoms of schizophrenia because of all the accumulation of these toxic molecules in the brain. Okay, and the brain sending signal: you have to sleep, you have to sleep, I need to protect, you need to protect. You maintain that person awake, terrible for the brain. So exercise, eat well, reduce stress doing exercise, maintain yourself active. That's one of the best things that you can do. Because it's proven that if we do all this, we can reduce about 50%, close to 50% of the dementia cases in the world. So we already have a treatment. We already have a way to reduce the cases, which is maintaining our hypertension, cholesterol, all these metabolic disorders. This was a study that was commissioned in Europe by Lancet, and they did a really comprehensive analysis of all the data that was out there, and they come up with the conclusion that we can, from 7 million here, people living with Alzheimer's disease in the United States, we can lower it to 3.5 just by having access to healthcare, living a healthy lifestyle. We can reduce those cases. With that, I then want to entertain your question. But before that, I want to acknowledge the current people in my lab and the funding that I received to conduct this work. So thank you so much.
Okay, please wait for the microphone with your questions. And if you, the title of this presentation was The Science of Alzheimer's. If you remember everything you heard today, you do not have Alzheimer's. So, having said that, let's have some questions.
There's a question right there.
Are there any medications that help?
Medications that help. Yeah, you see a lot of commercials out there about brain power or this and that, green tea, all these aspects, right? I would say if you feel good about it, there's nothing against it. I think that some of them, because they lack controls on how they are marketed, they usually have a placebo effect. And you buy it and you're spending that money and you're like, oh, it's helping me. This is the best thing. But it's actually a sugar pill. And we don't know, there's no control for these supplements of what actually they have. Ginkgo biloba, for example, was marketed as to reduce A beta, and clinical trials indicated that that's not what's happening. But if you have antioxidants like vitamin E, maintaining your vitamin B complex, especially vitamin B12, vitamin supplements, it will be the best one that I will suggest. Someone with with vitamin B12 deficiency can show symptoms of dementia. And just by increasing the vitamin B12 in the blood, the person come back. But if you don't treat it, then it's downhill from there. Yes, sir. There is a question. Oh, yeah. Yes.
Yes, I was just wondering, when you mentioned the women have 2 times the risk as men, does that even taking into account the fact that women tend to live longer and maybe would have a higher incidence as they get older?
Yeah. Those studies has been corrected by age. And what had been pushing more and more is about the menstrual cycle and menopause. So there have been contradicting reports, but in the past 2 years, I would say close to 5 years, there have been studies following women with early or premature menopause versus someone that developed at the age that they're supposed developing. And then using these tracers, these radioactive tracers in the brain of the woman. And they have found that those that are in premature menopause, without getting into hormone replacement therapy within 5 years, they tend to accumulate faster these pathological hallmarks that a woman that developed that had menopause at the average age and was put in hormone replacement therapy. There are other factors. There are social factors too, like the stress aspect. That's our fault too. We're promoting risk factors in women, but stress factors, especially in particular generations where the women were more toward stay at home or have lower education than men. There is an aspect that I didn't mention of all these related to education, is the theory that we call it cognitive reserve. So when you are in an enriched environment, it's like a banking account, like a saving account in the bank. You start accumulating this cognitive reserve, which means that you have these stem cells in the brain and you actually made the connection stronger versus someone that is in a stressful environment. That one will have less in their saving account than someone that are in an enriched environment. When these pathological molecules start accumulating, now you start taking from that saving account and you can mask the clinical presentation. There are people that die with the same pathological burden and never have clinical presentation, which is something that we don't know, we don't understand. How is that? What is the threshold to go and present? And the hypothesis behind that is cognitive research. Yes, sir.
So we hear about Alzheimer's now, and I'm a retired physician. When I went through medical school, there was no term Alzheimer's. It was always organic brain disease or organic brain syndrome or something like that. But the term and the definition and a lot of things you're talking about, it was not used 45 years ago in studying behavioral pathology. That's one thing. And so that's one question. Second thing is I think the women are more involved because they're just so much smarter in the first place. Now, if you start to see a small percentage of change, you notice it more quickly.
That's also a smart man right there. Yeah, you're right. Actually, I developed a course at the medical school at MSU that is called Molecular Neuropathology of Neurodegenerative Diseases. And we go from molecules to people living with the disease. And we talk about all the signals and the molecules and all that. And then we go to the pathology for brain structure function so that they can associate the symptoms. And then the last day, we bring people that are living with the disease that we're discussing in that week so they can see the manifestation of the symptoms. And we have very generous people that are living with these diseases, different tauopathies, Alzheimer, Parkinson, Huntington disease. Even we had one case of juvenile Huntington disease, and then because it's a mutation that affects a particular gene and pass through generations and increase anyway. But anyway, you can get it when you're a teenager. And she came and she was with less repeats in the 20s. She was in her 20s, but her sister was a teen that already had the disease, but she had less repeats. So she was expecting when she's gonna develop the symptoms, which is terrible. And she talked to the student, and then you see all the students crying, and it's really something that we're trying to bring to the physicians, those future physicians, those that want to go into neurology, to look at the individual and try to deliver this kind of diagnosis in a very empathetic way, because we don't have currently a treatment. And we tend to, there are unfortunately physicians that does the diagnosis and go fix your paperwork and talk to your family, and that's it. It's not that they're going to start dying right there. I mean, there's things that they can still be functional and do and be productive, and we should not dismiss them right away at the diagnosis. So, yes.
You said that during sleep it gives your body the chance to cleanse these things from your brain.
Mhm.
Is there any kind of urinalysis test that tells you how well your body is doing that?
Well, the short answer is no, but the reason why we know that that is happening is because it was tested. And what it was tested first in mouse models, in rodents, and then it was tested also in humans, looking at the CSF, at the cerebrospinal fluid. So they were doing this, following the circadian rhythm of the individuals and taking from the CSF samples and then measuring. And A beta is also clear during that process of sleep. So that proof of the biology was tested that way. But what you're going at, I think, is whether there is a differential, every individual has differential ways to clean that, and whether we can measure whether, for example, if you are better than someone or you're worse than someone, we don't have that kind of test. We know the biology that is happening, and that's why it's recommended. But the rate of clearing the brain is still, if it's different between individuals, I don't know. Yeah.
If you've lost track of time, we are well past 10:30. So if you have other plans today, feel free to leave. Are there
Up
any
to 11.
questions? Are there any questions from online? Okay. Any more questions here?
There is one.
You've talked a lot about the various aspects of the molecular searches for causes for the disease.
Mhm.
Let me focus on tau. It's present in all cells. Any cell that has microtubules has tau, I think. And is there something specific about brain tau that permits it to phosphorylate in ways that cause it to tangle?
Yeah.
If you look at, for example, at other tissues, do these tangles appear?
Yeah. So first of all, tau is mostly expressed in the brain. There have been some studies of what is called peripheral tau. That is a different form of tau than the one that we have in the nervous system. Is a longer tau, which also expressed in the peripheral nervous system. So our brain is different to other organs in the sense that express 6 different forms of tau. So from one gene, there is a process called selective splicing that generates 6 different proteins of tau. And the reason why I'm adding that piece of information is because one of the hypotheses is that affecting the ratio of those 6 isoforms is what could also promote the aggregation of tau. The other aspect is that we don't understand is that this tau aggregation, even though it's the same tau in all the neurons in the brain, it only aggregates in specific brain regions. And that specificity of the brain region is what also distinguishes Alzheimer's disease versus versus corticobasal degeneration versus progressive supranuclear palsy. That those three are characterized pathologically by the accumulation of tau, but they start accumulating in different brain regions. That's why they have also different clinical presentation. What is the selective, what contributes to the selective vulnerability of specific brain regions to initiate tau aggregation? We don't know. Well, yes, thank you for bringing that up. So one of the things that made tau to detach from the cytoskeleton is a modification, a post-translational modification that is called phosphorylation. But tau, physiologically speaking, requires to be phosphorylated to carry on this function. So is the aberrant phosphorylation of tau, which actually do the transition from physiological to pathological. What specific phosphorylation sites and what kind of kinase, which is the enzymes that add the phosphate group to the protein, are mediating that transition from physiological to pathological? There are some candidates that we know, but unfortunately, inhibitors of those kinases have more side effects than being beneficial because they're involved in other pathways that are required for the neuronal function. So we still don't know. Yes, but if we can go deeper, not only phosphorylation, there are other modifications, ubiquitination, acetylation, methylation, summorylation. There are different modifications that are found associated mostly with the pathological form of tau. How those modifications together promote that change is being understood. It's being studied right now. Yes.
Excuse me, we have a question online. Will this presentation / slides be available?
Yeah, it's actually saved here in this computer. You guys can feel free to share with anyone.
I'm curious, is dementia and Alzheimer's just a matter of degree?
No, dementia, let me explain that to you. Thank you for your question. So dementia is the term used when the physician don't really know what type of dementia is. Typically, it's a general term used because it still is not clear. I need to wait 6 months, a year, sometimes even 2 years to differentiate between frontotemporal dementia and Alzheimer's disease, for example. But then you have in your code for billing, you have dementia, and dementia type, Alzheimer type dementia, frontotemporal type dementia, vascular type dementia, so you have these different subcategories in the coding, billing coding for dementia. But yes, once you have, let me use your question to make the difference between mild cognitive impairment, which is MCI, which is something that people receive a diagnosis of mild cognitive impairment, versus dementia. Mild cognitive impairment is that you have, as the term indicates, just a mild problem with a speed of function, or either memory or processing information, so it's slower. But it's not to the level of affecting your daily life activities. It's not that it's affecting you to carry on with your life a specific function, like go to buy groceries and come back, or navigate your neighborhood, or remembering specific things, or making decision about paying your bills. Sometimes one of the things is that the children notice is that the person used to pay on time all the bills, and now I have 3-4 months unpaid bills, right? No, I did. I pay it, but is this affecting your daily life living? That's when you're making then the transition toward a dementia type of diagnosis. So that is very important to differentiate in terms of a cognitive dysfunction, that you still are working, you still are independent, you still have some issues in processing and speed, but you're still functional, versus that one that is affecting your daily life activities.
Anyone else?
I'm curious about your life and research. This is obviously very complex. Talk to me about your partnership with MSU AI department,
With
artificial intelligence department.
Well, yeah, talking about cognitive reserve and the use of, artificial intelligence. So right now, to tell you in terms of the algorithmic figure that I show you, putting this in a system that actually is learning as we incorporate this system, like machine learning, is very useful. Because then you're generating data as you use the algorithm, and the algorithm is learning how to modify and because one of the issues is thresholds. If you have, and I mentioned that A beta and tau can be measured even in the plasma, but it's not as specific or sensitive enough to use it yet in all clinical diagnosis, because when we do research, we know it's associated with Alzheimer's disease, because when we look at the level of A beta and tau together, we give a number in normal individual is here, but and then we have 1,000 individuals, right? The average at the range is here, but when we have individuals with Alzheimer's disease, then it's here, and statistically different. They are statistically different. But we have individuals with Alzheimer's disease that are at the normal level, and we have individuals with normal condition that are at the range of Alzheimer's disease, right? So then the problem is, what is the threshold? What is the difference between these individuals here and this one here? And we don't know what that threshold is. It might be that these individuals already have a beta, but have the cognitive reserve that I was mentioning, and that's why they don't have it. So we can actually have the system learn about that threshold and try to make better decisions about it. That's something how we can use artificial intelligence. Yeah, you have it.
Very much. It's been very interesting. I'm wondering other brain functions related to Alzheimer's incidence. For instance, all the hands in your question slide are all right hands. And so I was wondering if ambidexterity had any, or the lack of ambidexterity had any relationship to the percentage of people with Alzheimer's.
Well, thank you for that, because right-handed have been associated. It's something that we actually record in the clinical history is actually record whether you are right or left-handed, and it's associated more with right-handedness. But that's an issue by itself, because we have more people that are right-handed than people that are left-handed. So, yeah, it is something that we take in consideration, and neurologists take more in consideration when they are doing as an information to interpret the brain imaging, when they're looking at the brain imaging, because then if you are right-handed, then they're looking more at what is the deficiency in your left brain, because you tend to use it more, right? Or actually it's your dominant area. So they start guiding their way that they interpret the MRI and the PET scan based on the which one? Yes, that will help, similarly to speaking multiple languages.
Yes, exactly.
Similar to speaking multiple languages, if you can learn another language or you use another language, you switch back and forward. Again, it goes to that cognitive reserve property that you have the flexibility in the brain that goes back and forward. Yeah.
Thank you.
Hi, are you aware of any connection between autism and dementia?
Well, the difference is that one is neurodevelopmental and the other one is neurodegeneration. That's the main difference between them. In autism, is neurodevelopmental, we don't understand yet. It's not my area of study, but what we know is that the way that the brain is connected, that's why there is in a spectrum, because their connection doesn't follow the norm of the connections in the brain. And then you have this spectrum of individuals with autism, right? It's depending on how this, it's like having this building with all the cables, right? How it's connected. This building is different than the other building and the other building. So that brain is actually, when it was creating those networks, somehow got in a different way than the normal. And sometimes in that process, you have different expressions of the connectivity differences. But I don't, as far as I know, and I might be incorrect of this, there is not a direct association between autism, that autistic individuals that get to older age and a higher increase of dementia. I think that's what it goes to your question.
Yes. You were talking about cerebral spinal fluid as a marker or a way to assess, and the blood. I recently have read something about being able to look into the eyes, and I'm wondering what research is being done there to assess the degree of Alzheimer's, or what markers are there.
Thank you. I'm laughing because I always tell the program that I direct is to train the future neuroscientists. And I always ask these students to actually go and do presentations like this, because you really get tested if you know your thing. Because when we present in our community, I present EFHD2, and I can anticipate where the questions are coming from because it's very specific. But in here, it comes, when you are in the community, it comes from different areas. So thank you for saying that. Actually, there has been a lot of studies looking at inside the eye, because A beta, you can detect A beta aggregation in the eye. And they have actually a group in Harvard that actually are doing a stimulation of the eye with different wavelength as a therapy for the brain. But it's not at the clinical level yet. But yes, they're both for the diagnosis and also for therapy, non-invasive therapy of putting someone with this wavelength in the eyes, to stimulate specific areas in the brain to try to reduce the progression of the neurodegeneration in the brain. Yeah.
Anyone else? Irving, this has been a spectacular and well-received presentation.
Thank you.
I suspect that anyone that understood it all probably could take the biology or the biochemistry final exam here at Hope College and do perfectly well. So thank you so much for.
Thank you. Thank you.